Hi, I'm Jaron,
I'm Tyler.
and over the summer we looked at estrogen pollution and the feminization of fish in the Alawai. So this time we just wanted to thank Papa Jack for gifting the endowment that allowed us to do this research. Without it, we don't know what we'd be doing over the summer. So- and we also wanted to thank Dr. Yoshizawa for helping us with the histology slides, Dr. Maxfield, who took out time out of her precious Saturday to help us decode the histology slides, and Dr. Dulai for helping us and Maddie look at the estrogen concentrations. So the background. There are over 10,000 cesspools in Oahu, and 100 of them surround the Alawai and its tributaries. And so as Maddie said, there's some high levels of estrogen that have been measured around and in the Alawai, but the negative effects of this estrogen level has not been studied. And so our research was trying to look at if estrogen pollution has altered the wildlife living in the Alawai Canal, and if it has, how have they been altered? Our hypothesis was that estrogen pollution within the Alawai has negatively impacted the sexual function of its inhabitants, feminizing males and inhibiting the sexual function of females. So how does estrogen pollution occur? Maddie already kind of explained this, so we're going to review it. But the cesspools are basically like the alternative for the sewer system, except it's a less sanitary solution. So the sludge piles up on the bottom, and all the wastewater has the chance to leach out into the surrounding soil and even sometimes into the groundwater. And so on Oahu, we have quite a few cesspools, and especially around the Honolulu area where we have that big cluster. So we zoomed into the area around the Alawai Canal, and you can see, especially by the Makiki streams, there's a bunch of cesspools that are around there that could potentially be leaching the waste into the Alawai. And so Maddie's study, again, it looked at the levels, and her highest concentration was 30 nanograms per liter, while we see effects that are even lower than that. The 30 nanograms per liter actually was just one day, one time, and so the rest weren't as high as that necessarily, but that was still one of the measured concentrations. And so you can see rainbow trout at 2 nanograms per liter saw a decrease in the rate of testicular growth. The fathead minnow at 4 nanograms per liter saw the sex ratio skewed at 84:5 of male to female. And of course, the tilapia, which we looked at both 5 nanograms and 25 nanograms per liter. After 10 days, the sex ratio shifted and the male's weight was significantly decreased. So the synthetic estrogen we looked at, EE2, is like one of 4 major groups of the estrogen family. Estrogens include like estrol, estrone, estriol, esterol, and of course estradiol, which is like the biggest of the 4 in terms of affecting wildlife. And so it comes from these oral contraceptives where when a person takes them and their body uses what it needs, it sends out the waste in the form of urination and it goes into the cesspools. And so the unique thing about estrogen, the reason why we can with some certainty say that it's the estrogen doing the change in sexualization is because it kind of has its own special reign over the female sexualization, and it plays a role in hormone production, gonadal development, and body weight. And a big misconception is that it only affects males, not females, but it affects both. It hinders the ability of both males and females in their sexual development.
So these are the procedures we used during our experiment. So first off, the fish that we examined were, as Jaron said earlier, tilapia. So we looked at 2 species. One was the Nile tilapia, which is mainly from aquaculture, but is also present in the Alawai. But the bigger species that we looked at was the black shrimp tilapia, or Saurotherodon, which hails from West Africa. And this species of tilapia is the dominant presence in the Alawai because of its ability to tolerate higher salinities. So we looked at these 2 species during our research. So in order to determine if the estrogen in the Alawai has had an effect on these fish, we looked at 3 things. So one was the sex ratio between males and females. The second thing was the gonad mass to total mass ratio, also known as the GSI. And the third thing was histology slides of gonad tissue that we extracted. So in order to gain our data, what we did is we dissected the fish using scientific tools such as the scalpel or the forceps over there, and we extracted the gonads, which we masked using a balance, and we also masked the fish to see its relative ratio, and we placed the extracted gonads in a 4% paraformaldehyde solution, which we sent off to UH to make histology slides. We also took fin clips or flesh samples from the fish in order to determine species for certain. So we would do a PCR reaction using the CO1F primer or cytochrome oxidase 1, which is part of the mitochondria, and we sent that off to a lab in California to completely sequence. So you can see the nucleotide sequence on that far picture over there.
So these were our findings, like the fun stuff. We looked at the sex ratios first, and according to this Shved paper from 2007, they looked at the female and male bony fish, and you can see on the right there's the control sample, and on the left there's the experimental sample, and you can see clearly that the female population was much bigger than the male population in the experimental section. And so this is what we found. The undetermined parts of the pie charts were because the fish were too small, so either the gonads were too small or we couldn't identify whether they were male or female. But other than that, you can see in the aquaculture tilapia, even though our sample size was quite small, that the males and female, the ratio is pretty much 50-50. Whereas in the alawai tilapia, we see 75% male and just 10% male, which is a big difference. And so we inputted our data into a Fisher's exact test. We got a value of 0.0077, significant at p less than 0.05, which is basically, we can be sure that this extrapolates to the entire population like 95% of the time. And so the second thing we looked at was the gonad to mass ratios. But unfortunately, if you can see the error bars, they're huge and they overlap. And so the data is not statistically significant. But even though that happened, it's still interesting to see how the alawai compared to the control fish, the males, for the alawai, it was about like 3 times the percentage as the control. And for females, it was the opposite. The controls were 3 times the percentage of the alawai. And so the third thing we looked at was the gonad histology. So basically we got the gonads and we sliced them up, but we didn't do that. We sent it to UH and Dr. Yoshizawa did it for us. And so we looked at male and female histology. The female histology, if you see, both A and B show different samples. In A we have healthy ovaries, and in B we have ovaries from an EE2 amended late. This is from KID 2006, a different study. And so we tried to mirror what they looked at, but in our tilapia. So you can see on the left side with the healthier ovary, there's like 1.5 of these follicles that look weird. They're called like atretic follicles. And follicle atresia is important to the function of ovaries. It's basically clearing out the old follicles so that new ones can grow and develop the oocytes. But then if you look on the right, we have atresia-ridden ovaries, and they look much uglier, and there's lots of space in between, much more treated. And basically the healthier ovaries, the fish that it came from, was about like 1/6 the size of the atresia-ridden ovary sample. And so although they're both from the alawai canal, we think this could be because the bigger fish has lived in the alawai for longer, and so the effects were more evident. And so in the male histology, we looked at the same study, KID 2006, and they found oocytes in the male testes tissue. C is the healthy testes tissue, D is from an EE2 amended lake. And there are these growing, developing oocytes in the remnants of previously degraded testes tissue. And so basically the male gonad is now producing eggs. So in our samples, On the left, there's healthier testes. This is from the controlled sample. In the middle, you can see all the healthy spermatogonia. But on the right, from the olive tilapia, we see two possible developing oocytes. And so in the male tilapia from the olive, there are these egg cells growing in the testes tissue.
So after looking at all these results from our data, what does this mean and why does it matter? So the conclusions we can draw from this is that tilapia may have been feminized by the presence of estrogen in the olive. We can determine this because the sex ratio has been skewed towards more females, and some gonad samples showed signs of degradation, which helped to support our hypothesis. So as such, we can assume that reproductive health of these fish should be questioned. But even though we've seen these results, our sample size was small, so we can't really draw any definite conclusions as of right now. So why does this matter? It could also affect other organisms, including native species or commercial fish living in the alwai or by the area in which it dumps into the ocean. We could also see that other pharmaceuticals could also be leaching into streams, and antibiotics released in this way could bring about antibiotic-resistant bacteria. And even though the concentration of estrogen was variable in the alwai, we still saw some effects from it. So if antibiotics were to do the same, who knows what the effects could be on bacteria. So, thank you.
Oh, it took out the other.